Friends, welcome to Biology Made Easy. So we are discussing mutation. In the previous video, we have discussed the different types of mutagens, how mutations are classified, and again we also have discussed what are the different types of aneuploidy. So today in this video, we will discuss the remaining part, that is euploidy and also chromosomal aberration. So let us start with the euploidy. So in euploidy, what happens? The chromosome sets are increased. Suppose in a haploid set, the chromosome number is n. Now it will be multiplied to 2n, it is diploid. If it will be 3n, diploid. If it will be 4n, then it is a tetraploid like one. If it is the genome is A, then this is double A, this is triple A, then this is tetraploid A. That means the same genome is multiplied several times, then this gives rise to euploidy. Usually, in case of euploidy, which again divided it may be, if it is only one set, it is monoploidy. If there are two sets are there, it is diploidy. But if 3, 4, 5, 6, 7, 8 like are there, then they are known as the polyploids. That means this is haploidy, this is diploidy, and all these they are coming under polyploids. Now our main focus is to discuss about these polyploids, because normally all organisms are diploids. So how these polyploids are formed? Usually polyploids are formed by applying the chemicals which may be colchicine or it may be garmosan. Because these are the two chemicals which inhibit the spindle fiber formation during mitosis. So when during mitosis, when the chromosomes separate to the poles and the spindle fibers draw them, these spindle fibers are not formed, so that is why the chromosome number is doubled. Suppose in one organ 2n is equal to 24. Then the gametes should have 12 if they will separate. But suppose they will not separate. What will happen? If they will not separate, then this will be there together. So if 24 chromosomes will not be separated into their chromatids, then ultimately the number will be 48. That means if this is diploid 2n, then it becomes 4n tetraploid. So in this way the polyploids are formed, and the polyploids have a more number of chromosomes. That is why the size of the nucleus becomes greater in size. The organisms are more gigantic. Usually the polyploids are more common in case of plants than animals. So usually the polyploids are very gigantic, their leaf size, flower size may be bigger. But here one thing is very important. When we increase the ploidy level from 2n to 3n, 4n, 5n, 6n, the size of the different parts increase. So one may think that when the size increases, let us increase the ploidy level to such an extent that the size of all fruits, grains will be very bigger. But no. Why? Because each plant has one ploidy tolerance level is there. Some plants can tolerate 4n, some can tolerate 6n, 8n. That is the limit. That is why you cannot increase to any level. That limitation is there. So polyploids usually play a very important role because the polyploids are usually fertile. That is why they play a very important role in agriculture. Now the polyploids, they can be classified into autopolyploids, allopolyploids, and autoallopolyploids. These can be classified into autopolyploids, allopolyploids, and autoallopolyploids. Usually In case of autopolyploid, this same set of chromosome is multiplied. Suppose the one set is A. Now if it will be 3, then it is known as autotriploid. Autotriploid. If it will be 4, then it will be autotetraploid. Autotetraploid. So the common examples of this autotriploid is seedless watermelon, and another example is cynodon-dactylon, that is commonly known as dub grass. Cynodon-dactylon, which is commonly known as dub grass. They are all autotriploids. Now, what are the examples of tetraploids? Common examples are usually grapes are there, banana, and sugar beet. These are the common examples of autotetraploids. So, since they are fertile, these are fertile, so we will get some benefit from them. Now, come to the allopolyploids. Allo means different. So, when allo means different, usually these are obtained by hybridizing two plants and then multiplying the chromosome number. That means if we take two plants, one is AA, the other is BB, and these two plants have some good characters, and we want to hybridize between them so that the F1 will have better characters. So the F1 will be AB, but this is sterile. Now, if the chromosome number will be doubled by applying colchicine, then it will be like this. That means A in double and B also in double genomes. Now, this plant is fertile. Because the chromosomes of these two can pair and chromosomes of these two can pair with each other, that is this fertile. Now, this plant body has two different sets coming from two different plants. So, this is known as allotetraploid. Allotetraploid. So this allotetraploid is more useful than autopolyploids because here we can combine the characters of two plants and then we can double the chromosome number so that we can get the benefit as well as the plant is fertile. So some of the examples of this allopolyploids are, one is Raphanobrassica. Raphanobrassica. In case of Raphanobrassica, the Raphanosativus is radish, and here the chromosome number is 18. Then it was crossed with Brassica oleracea. Here the chromosome number is also 18, and both of them also belong to the same family, that is Brassicaceae. When these two are crushed, then it will be RB and 18, but this F1 hybrid will be sterile. Now by applying colchicine, if it will be doubled, then R will be doubled, B also be doubled, and the chromosome number will be 36. So see, this is a allotetraploid having the characters of both radish as well as cabbage. So this hybrid was made by Russian scientist Karpencheko with the idea that in the same plant the radish will be underground and the cabbage will be above. But unfortunately, the reversed characters came. That means the upper part of radish and the lower part of cabbage. So economically it was not successful, but a new plant was evolved, that is Raphanobrassica. Similarly, we can take the second case, that is our cultivated wheat, that is Triticum aestivum. So Triticum aestivum was obtained by same method. Triticum dicocoides, it has usually 4n number, and 4n is equal to here 42. This was crossed with Aegilops squarosa, and here 2n is equal to 14. So now the F1 hybrid 2 will have here from here 21 chromosomes, from here 7. So the plant will have This is 21, this means 28 chromosomes, and it will be sterile. Now, when with the help of cultivation it was doubled, then the new plant has 56 chromosomes. Sorry, this is 2n is equal to 28, 2n is equal to 28, so it will have 21, and this will be 42, and this is Triticum aestivum. So this is Triticum aestivum is an allohexaploid. So this is the common wheat that we are cultivating. In the same way, the tritical plant was also obtained. Tritical, it is the first man-made cereal. First man-made cereal is tritical. So this was obtained either by crossing this one with the sickle cereal or this one with the sickle cereal. So if it will be obtained by crossing, suppose this is Triticum aestivum. So Triticum aestivum, here it is 6n is equal to 42 and crossed with the sickle cereal, and here 2n is equal to 14. So it will have here 21, here 7. So it will have 28 chromosomes, and this is sterile. Now if it is doubled, then it becomes 56. So it is 6n, that is 2n. So now this is octoploid, and this is known as tritical. The first man-made cereal, tritical, was obtained by this method. So likewise, tetraploids. This is allohexaploid. This is allooctaploid. So in this way, we can get different types of allopolyploids, and allopolyploids are more important for the purpose of agriculture, where we can combine characters and again we can multiply it. Now coming to this autoallopolyploids. In autoallopolyploids, there will be characters of both autoploids as well as allo. That means the autoallopolyploid, one example is this is the auto. Now this is allo. So this is known as autoallopolyploids, and common example is Helianthus tuberosus. So these are the different examples of this autopolyploids, allopolyploids, and autoallopolyploids. So this is about the different types of euploidy, from which we can get monoploidy, diploidy, and polyploidy. And the polyploidy are again divided into autopolyploids, allopolyploids, and autoallopolyploids. So this is about polyploidy. The next part, that in the mutation, we'll discuss chromosomal aberrations.